Hi everyone, my name is Josh Scallan. I'm an assistant professor in the Morsani College of Medicine at the University of South Florida. I'd like to thank the organizers for inviting me here today to present our research. And although we just recently got into studying lymphatic anomalies about a year ago, my laboratory is probably more well known for pioneering unique methods to study various aspects of lymphatic function, including the ability of lymphatic vessels to contract and their permeability. And that's going to be the focus of my talk today. So before I begin talking about various aspects of lymphatic function, though, I just wanted to point out that lymphatic anomalies is a collection of diseases that have very different presentations, and they can look very different, so they're very heterogeneous. And the main way that I think about lymphatic anomalies is that I ask the question of whether lymphatic function is altered inside the lesion, and how does the lesion affect lymphatic vessel function. So we can talk about different types of lymphatic vessel functions. One of the functions of the lymphatic vessels I've already mentioned is their ability to contract or pump lymph downstream, so they're not just passive tubes. Another type of function that our lab is primarily concerned with is measuring the permeability of lymphatic vessels to various solutes and what can leak out. And then finally, another area that we've become recently interested in is how lymphatic vessels grow and maintain bileaflet valves. So I'm going to walk you through how our laboratory measures all these various functions. So with lymphatic contractile function, we usually surgically excise a lymphatic vessel from the mouse leg. That would be the popliteal lymphatic vessel that runs parallel to the saphenous vein. And once we've excised the vessel, we clean it, and we tie it onto two small glass tubes. And then with the two small glass tubes, we can control the pressure inside the vessel. And we transfer this entire apparatus to a microscope outfitted with some special software that can detect the vessel edges and measure the vessel diameter over time. Here's what that looks like on a graph of diameter versus time. Each time the vessel contracts, the diameter very abruptly and quickly decreases the diameter, and then when it relaxes, the diameter goes back to where it began, so you get these series of peaks here. And we can extract a lot of information from these peaks, and one thing you may notice is that as we increase the pressure in the lymphatic vessel lumen, the vessel has to work harder to contract, so its contractions are smaller and smaller. So like I said, we can extract a lot of information from this kind of graph, and we can describe various aspects of lymphatic vessel contractile function, such as how strong the vessel can contract, or how frequently it can contract, or maybe even the ejection fraction, how much volume it can pump with each contraction. And we can use this information to then diagnose problems with lymphatic smooth muscle cells in various knockout mice. The other type of function that my lab cares about measuring is lymphatic vascular permeability, and we have a very similar assay to measure that. So the way this works is we measure the total amount of light in a region of interest, in a rectangular region of interest, and we'd use a photometer to do that. A photometer is just a tool that measures photons. So without going into too much details with how this works, we can perfuse different fluorescent dyes or molecules in the vessel lumen, and then over time these molecules are going to escape from the vessel itself and accumulate in the surrounding bath solution. And the important thing from the photometer recording that we care about is denoted by the letter C at the top, which is a linear slope, which is the rate at which the solute leaks out of the vessel. And so we can take the information from the photometer recording and we can calculate the permeability of the vessel at different pressures or to different molecules with different sizes. And we can wash the dyes out of the lymphatic vessel and put on inhibitors for certain pathways to try to probe what signaling pathways regulate lymphatic vessel permeability. We can also do heat maps of the lymphatic vessel as dyes leaking out. This is more qualitative and lacks quantitative power of measuring permeability, but it's useful to try to figure out where vessels are leaking from. All right, and then the last thing that we care about studying in my lab is valves. So we became interested in lymphatic valves because we wanted to study a protein that regulated permeability called VE-CADHERIN. So this is vascular endothelial cadherin, and it's a junction protein that regulates blood vessel permeability, but it's also expressed in lymphatic endothelial cells. But before we decided to study it. Little was known about how VE-CADHERIN regulated lymphatic function. So we made knockout mice to study VE-CADHERIN, and hopefully what you can notice from this is that if you look at a reporter mouse where all the lymphatics in the reporter are green, there are a lot of valves, and that's in the controls. But if we knock out VE-CADHERIN, we can completely inhibit valve formation in the embryo. If we do something similar postnatally, after a lot of valves have already formed, hundreds of valves have formed, you can see all the valves on the left-hand side as little bright GFP dots. And if we knock out VE-CADHERIN after valves have formed, they go away and regress. And this is nearly complete. Almost all the valves go away. And the reason that they do this is because we discovered that VE-CADHERIN allows the lymphatic endothelial cells to sense shear stress and lymph flow is what causes the valves to form, but also is required for the valve to be maintained throughout life. The other thing we noticed when we knocked out VE-CADHERIN was that the lymphatic vessels became really leaky. So at the bottom in panel D, you can see a lymphatic vessel that's white because it's absorbing lipid from the gut. But in panel C, in the VE-CADHERIN knockouts, the abdomen fills up with this milky fluid, also known as chylous ascites, which is reminiscent of several congenital diseases. And the reason that occurs is because the lymphatic vessels become leaky, as in panel E. So we became interested in lymphatic anomalies because we started studying KRAS G12D mutations. So recently, there was a paper published by Mike Dellinger and Mika Vicula identifying for the first time KRAS G12V mutations in Gorham-Stout disease. This is very exciting because what they found in this paper was that, among many things, is that when you induce the same mutation in mice as in the patients, a lot of the valves go away. About 90% of the valves go away, which is about what we saw when we deleted VE-CADHERIN. So at the same time that Mike Dellinger was studying KRAS, we were also studying KRAS in my lab, and now we've begun collaborating. But what we found out in my lab is if you take lymphatic endothelial cells and culture, and you infect them with KRAS G12D, so that every single LEC expresses the KRAS G12D, if you look at the middle panel, you can see that VE-CADHERIN expression is almost completely abolished, which was very exciting to us. And then we decided to see if we could confirm this in vivo, so we made vascular endothelial specific KRAS G12D mutant mice. And if you look at this staining of VE-CADHERIN on the left-hand side, you can indeed see that VE-CADHERIN localizes to the cell-cell junctions as it should. But when you have KRAS G12D being expressed, it looks a lot blurrier and hazy. And that's because we think VE-CADHERIN is diffusing away from the junctions or being internalized and degraded over time. And so although this doesn't perfectly look like the in vitro data, we think it's a very progressive thing that happens over time. So what we're going to do in the future is follow up on this finding by analyzing the valves and seeing if we can correlate the loss of valves with the loss of VE-CADHERIN over time. We're also going to apply our methodology that I talked about in the beginning of the talk to see whether or not KRAS G12D mice have any defects in lymphatic permeability or contractile function. Okay, so with that, I'd like to acknowledge everyone who's responsible for doing the work. Most of the permeability measurements that you saw were performed by my postdoc, Melanie Janaway. I'd like to also acknowledge Ying Yang, who is a researcher here who contributed a lot to the VE-CADHERIN paper, and I'd like to thank my various sources of funding who's kept my lab going. Also, I'd like to thank all the external collaborators who've helped us along the way, many of whom have given us new mouse models so we can perform studies or helped us with experiments. And I'd like to specifically acknowledge Mike Dellinger. We're very grateful to be collaborating with Mike on the KRAS project. So with that, thank you for your attention, and I'd be happy to take any questions you may have.